As a brief disclaimer, this video focuses on the ethics of genome editing in somatic cells, not germ cells. The SCGE program does not support any research activities on genome editing in germ cells. So, could you start by telling us your name, your institution, and your role?
Sure. I'm Ryan Spellecy. I am from the Medical College of Wisconsin, where I'm a professor of bioethics, and I also direct the Human Research Protection Program, which includes the IRBs at MCW.
And then, what's your role within the SCGE Consortium?
So in the consortium, I work primarily on ethics and outreach. In our group, I work on research ethics and outreach.
So, you're going to talk today about the research ethics of somatic cell genome editing in animal models. Could you
Right.
start by talking about the 3 R's of animal welfare?
Sure. So the 3 R's are sort of our ethical framework for thinking about research involving animals. And we start with the foundational premise that the needs of animals matter, but the needs, and in our context, the health needs of human beings take priority. And so that really gives us this framework of the 3 R's. There's other approaches in ethics, the beliefs that people have that would say that no, the needs or the rights even of animals are absolute. And so research on animals is never ethically justified. We're starting at a point where we think that research on animals is justified to further the health needs of human beings, but that's not absolute in the sense that we can just do whatever we want with animals. And the 3 R's are really how we think about that and the ethical framework for research involving animals. And the 3 R's are replacement, reduction, and refinement. Replacement says that we should replace animals or animal models with non-animal models whenever possible. Again, going back to that touchstone of we're trying to improve human health here. And so if we can still achieve the goals of the research with a non-human animal, we should. Or sorry, a non-human model, we should. And then reduction is really paying attention to we should use as few animals as possible. So what are the changes to our study design that we can make to reduce the number of animals? And it's actually an ethical concern in our work because some of this research requires a lot of animals to be used. And so it's something that we're always in tension with. And then the last one is refinement. And that's that we should refine our study design, our practices, our husbandry, all of that to reduce and minimize animal pain and distress. So replace, reduce, refine.
Great, thank you for that overview. Do animal welfare considerations vary among species, such as mice versus non-human primates? And if so, how do they vary?
Yeah, they do. Some of the concerns are that non-human primates, for example, different non-human primates, take longer to grow and raise and things like that, and so that slows the pace of research. One of the reasons we look at non-human primates in specific is for neurological disease research on neurological diseases. Like we might be looking at things like Alzheimer's or Parkinson's or something like that. And the reason it's sort of a double-edged sword. The reason we want to look at non-human primates is because their neurological systems are more similar to human neurological systems. But that also creates problems because that can result in increased pain and distress in the animals, which is one of those, the refinement, that's one of the R's that guides us. So there's a tension there. And there's even been some discussion in the literature about, say, research on autism, in which you would uh, use this sort of technology to create an animal model in which you would essentially create animals with autism, and one of the sequela of autism is self-harming behavior. And so you would be intentionally creating an animal that would in turn engage in self-harm. That doesn't mean that it's impermissible because we do this, think of oncomice and things like that. Those are animals that are created in order to uh, develop a cancer. And so that too has a pain and distress inherent in the model. But it does create challenges for, okay, if we're going to do that and we're going to create this animal model that part and parcel of the animal model is the likely potential for self-harm, how do we refine our practices? How do we take care of these animals to minimize the distress and the pain?
Okay, great, thank you. The next question is, do humans feel better if they know a product has been tested in non-human primates and was found to be safe?
So this is an interesting question, because there's sort of two ways, as I read it, two ways to take this. One is the simple question, do humans feel better? Which is really almost an empirical survey question. Let's poll people and find out. But I think the more interesting question is, should humans feel better if they know that a drug, for example, has been tested in animals and found to be safe and effective? And I think the answer to that is yes, although it's a qualified yes, because FDA is still going to require that that drug be tested in humans to be found safe and effective. So, in a sense, if we're talking about if I'm going down, if my physician writes me a prescription, should I feel better that it was tested in animals? Well, yeah, but I put more stock in the eventual clinical trials that show that it's safe and effective in humans, because one of the common criticisms of animal models is that not everything that happens in the animal model translates over into the human model in terms of both efficacy and safety. But if I'm, say, a person that's considering participating in a clinical trial, so that's a drug that hasn't been approved by the FDA, hasn't been shown yet to be safe and effective in people, I can make a better informed decision as to whether or not I want to participate in that clinical trial based on the animal studies, based on the animal models, because that will show us, well, these were the common side effects that we saw in animal studies. And so that gives us a sense of what we want to look out for and what I can expect as a participant, but also it informs the researchers doing the clinical trial that say, all right, if this drug showed liver damage in animal studies, well, then we know that we want to design our study in such a way that we're paying attention to that and we're looking for and anticipating possible liver disease or liver damage. And so we're going to monitor more frequently for that. So clinical trial participants should feel better too that these are studied in animals.
Great, thank you for that answer. The next question then is it ethical for the FDA to require animal testing for every product, even if a very similar product has had testing done and was found to be safe?
Right. So great question. And the short answer is yes, but I want to walk you through how we get to that yes. And again, it comes back to these three R's. So what we're really asking here is a question of replacement. So if a similar drug, similar device, similar cell editing process has been shown to be safe and effective, why do we need to test it on animals again? And the answer goes back to that question of, well, human health needs take priority here. And at the moment, to FDA's credit, they are actively and aggressively pursuing non-animal models and options other than animals for research for a number of reasons. One of them is one of the three R's, replacement, but also some of the things I mentioned earlier about that animal models are not without their problems in inaccuracies and things like that. And so is it ethical? Yes, because we haven't gotten to a place yet where, according to FDA, we can safely and reliably rely on these non-animal models. But again, we're working on it, we're trying to get there. And I would anticipate that as the research continues on these non-animal models, that we will start to see their use and more of their use. It would be unethical if those models were ready and we still used animals. That would be unethical. That would violate that replacement principle in the three R's. But we're not there yet, which is why it's still ethical.
As other testing options such as organoids become validated and available to researchers, how does this change the ethics around using the animal models?
Yeah, we go back to replacement, and we want to be careful, right? Because we want to make sure that these non like organoids and things like that meet the needs for the scientific question at hand. But if all things being equal, if we have an organoid or some other non-animal model that or non-animal option, if if they perform adequately, we should phase out the animal research in that area. This isn't going to be something where, oh, this technology is ready and now we don't need animals for research anymore. That's not the case, but we'll start seeing movement away in specific diseases, specific scientific areas of inquiry.
And then lastly, is there anything else that you would like to say about ethics in somatic cell genome editing?
Yeah, so in somatic cell editing, I think the one thing that strikes me about this is it's an exciting time to be involved in this, because I just thought back to, when things like CRISPR were first discovered and announced, wasn't too long ago, and I can recall giving a guest lecture in somebody's class about this, and it was all very theoretical, and we talked about, well, you might be able to insert something into this gene that would say, well, yeah, in a sense, cure sickle cell disease. But there are other aspects that we know and don't know about, what certain genes do. And so if we knock out a gene, or we edit a gene, there will be other effects because genes do multiple things. And we've gone from a place where we were talking about things like sickle cell and malaria and the effects that it has, that the sickle cell gene can have a protective effect against malaria. And so you have to kind of balance malaria versus sickle cell, and others as well, like HIV and things like that, that we could edit. We could edit the genome so that you would be less susceptible to HIV, or the virus couldn't implant on a cell and things like that. They were theoretical, we were thinking about them, to now in my role in the IRB, I see studies that involve things like CAR T cells almost every couple of weeks. Where CAR T is a really effective therapy for people who simply have no more options, and this can cure their blood cancer or something like that. And now we're starting to see it in other diseases as well, in neurologic diseases. And yet, as we know from the research, there are problems downstream with things like CAR T, where we have secondary malignancies that develop. And that kind of seems to be the theme to me with things like CRISPR and the like, is, boy, the pace is rapidly evolving. So I'm grateful that we're having conversations about the ethics involved in this today, because I think that's very important, that as the pace quickens, we keep asking these ethical questions and make sure that as scientists, as researchers, and as a society, that we're comfortable and we know the facts around these, and we're comfortable ethically with where we stand. But also, the other theme that seems to evolve with this is that there's always a secondary effect. And so we always need to be vigilant and looking out for those, whether it's an immediate effect based on the editing, or those downstream effects like we see in CAR T with secondary malignancies. All right, cool. Bye-bye.